Hello, my name is Carbonell-Rozas and I am a postdoctoral researcher in the field of green analytical chemistry. In this video, I will present the solid-liquid extraction of a wide range of multi-class pesticide residues from different food commodities, including spinach, wheat, and orange. For that, I will use Nades, or natural deep protective solvents, as a green extraction solvents. Firstly, I will show how to prepare the Nades and then his application in the sample treatment procedure. In general, the materials and reagents required for this procedure are quite easy to obtain. These include the main component of the Nades, falcon tubes, pipettes, and vials. Two solutions were prepared in advance: 1 molar sodium hydroxide, a 0.1 molar phosphate buffer. For the preparation of the Nades, the main components, thymol and menthol, were weighed using analytical balance. Based on a 1:1 molar ratio, the amounts were calculated according to the desired final volume. In this case, I only prepare a small volume, but the procedure can easily be scaled up if needed. The solid components are placed together in an amber glass bottle with a magnetic stirrer using the stirring and heating method. The temperature should be checked periodically to prevent exceeding the recommended limit. In this case, heating at 60 degrees Celsius for about 30 minutes was enough. After that, the EUTECTIC mixture is formed, resulting in a transparent and homogeneous liquid solvent. Then we need to prepare our samples, in this case spinach, wheat, and orange, which represent different groups of food commodities. First, the samples are homogenized using a blender to obtain an uniform matrix before extraction. After that, the homogenized samples are weighed into 50 ml falcon tubes, 1 gram for spinach and orange and 0.5 grams for wheat. In order to facilitate mixing with the Nades, water was added to the samples. For spinach and orange, pH adjustment was also necessary, so phosphate buffer and sodium hydroxide were added to create basic conditions that favor the extraction of the target pesticides. For wheat, only water was used. In all cases, the final added volume was 2 ml and the samples were vorticed for 10 seconds. Subsequently, 2 ml of the Nades were added to each falcon tube. Due to the high viscosity of the solvent, it is important to ensure that all the Nades is fully transferred from the pipette tips. Then the samples were agitated using a rotatory shaker for 10 minutes and centrifuged for another 10 minutes at 4,500 revolutions per minute, preferably under cold conditions. After this step, phase separation was successfully achieved. From the supernatant corresponding to the Nades phase, and a liquid of 0.5 ml was collected using a pipette, transferred to a vial containing the same volume of methanol, and vorticed to ensure proper mixing. The vials were then analyzed by HPLC triple quadruple mass spectrometry, operating in positive electropionization mode under multiple reaction monitoring conditions. Here we can see a schematic overview of the solid-liquid extraction, which summarizes the procedure. This methodology was then validated according to the SANTE guidelines for pesticide analysis, considering linearity, precision, limit of quantification, and recovery. Although this method was initially optimized for 11 representative pesticides, it was later validated for a larger number of compounds, including those commonly investigated in routine pesticide analysis, reaching around 250 compounds, for example, in the case of spinach. The sample treatment was also evaluated using the AgriPrep and Vagi metrics to assess its greenness and practicability. In both cases, the results were very positive, highlighting not only the sustainability of the proposed procedure but also its potential transferability to other laboratories. As concluding remarks, I would like to say that this study demonstrates the feasibility of using Nades as a sustainable alternative to conventional organic solvent for multi-class pesticides analysis in food safety. The Nades used in this study, composed of thymol menthol in a molar ratio 1:1, provided satisfactory recoveries and acceptable matrix effects for a high number of compounds, as well as compatibility with the chromatographic separation and mass detection. Thank you for watching. If you would like to learn more, feel free to contact me or follow my work on these platforms to stay updated on my latest research and results.